Hello everyone, my name is Anastasia Antoniou and I'm a PhD candidate at the Cyprus University of Technology. The current study concerns the development and evaluation of two simple 3D printed robotic systems for focused ultrasound-mediated opening of the blood-brain barrier in small animals. So two robotic devices were engineered to facilitate opening of the blood-brain barrier in mice and rats. Both devices were manufactured on a rapid prototyping machine and comprised a single-element spherically focused transducer with a nominal frequency of 1 MHz. The first version features two horizontal degrees of freedom, where motion is actuated by piezoelectric motors and controlled by optical encoders. So the positioning mechanism and the ultrasonic transducer were arranged into this compact enclosure, which includes a dedicated platform with adjustable handles on the top to immobilize rodents of different size and type above the transducer. The second version of the device has a completely different design with a motorized vertical axis so that ultrasonic energy is delivered from top to bottom. Specifically, the transducer is housed into this conical structure whose height is adjusted. Following ex vivo assessment of the developed systems, feasibility experiments were conducted in wild-type mice in collaboration with the Cyprus Institute of Neurology and Genetics to assess their functionality in terms of blood-brain barrier disruption. Regarding the experimental procedure, initially the mouse head was shaved. The mouse was then anesthetized and following an intravenous administration of 5 microliters of sonovoid microbubbles, and then positioned the device and received a single sonegation at 1 MHz using 10 millisecond pulses at a duty factor of 1% and electrical power of 20 to 50 watt for a total duration of 100 seconds. Evans Blue was injected intravenously immediately after sonegation and about 35 minutes before the mouse was sacrificed. In the first version, the mouse was mounted on the dedicated platform shown here, and the container was filled with degassed water up to the mouse head for proper ultrasonic coupling. Whereas in the second version, a top-to-bottom approach is used, where this water-fill cone is coupled to the target using ultrasound gel. In total, 10 mice were enrolled in the study. One mouse served as the control mouse and received no treatment or Evans Blue. One mouse received Evans Blue only, and 8 mice were treated with focused ultrasound plus microbubbles. Assessment of the blood-brain barrier disruption was based on the well-known Evans Blue dye method. Immunostatin with the fibrinogen and fibronectin antibodies was also conducted to assess the leakage of the plasma proteins into the brain parenchyma. Representative microscopy photos of Evans Blue extravasation in the brain parenchyma adjacent to the lateral ventricles are shown here. So as you can see, no leakage is observed in the brain parenchyma of the control mouse and the mouse injected with Evans Blue only, whereas Evans Blue leakage is evident in the mice treated with focused ultrasound. Note also that with increasing acoustic power, we have higher levels of Evans Blue dye in the brain tissue covering a larger area. The BBB permeability was also characterized using fibrinogen and fibronectin immunofluorescence staining. The mice treated with focused ultrasound plus microbubbles showed higher levels of the protein in all examined brain areas compared to the control mouse as expected. For example, images of fluorescent microscopy from the corpus callosum are presented here, where the fibronectin is stained green and the cell nuclei are stained blue. In the case of the FUS-treated mouse, there is protein leakage and distribution throughout the examined brain tissue. So blood-brain barrier opening was achieved with both robotic systems. However, the first version presented some difficulties in accommodating the anesthesia needle. This issue was addressed and solved with the proper design of the second version. In addition, with the second version, there is no possibility for water leakage, and it also enables visual confirmation of proper acoustic coupling. On the other hand, an advantage of the first version is that it is also suitable for medium-sized animals such as rabbits. In conclusion, the proposed devices constitute a cost-effective and ergonomic solution for focused ultrasound-mediated opening of the blood-brain barrier in small animal models. The follow-up studies will assess the efficacy of delivering various chemotherapeutic drugs through the opened blood-brain barrier. The study was funded by the Research and Innovation Foundation of Cyprus under the project SoundPad. Thank you very much for your attention.